Thank you for the introduction, and thank you for the organizers for the invitation to speak to you today about what is actually a new program in my laboratory, developing over the last 3 or 4 years. And I've been very fortunate at Michigan State to be part of a large group of faculty, graduate students, and postdocs who are all interested in bovine leukemia virus, which, as I hope to convince you in this talk, has become a serious issue in the United States, Canada, Japan, Argentina, in fact, much of South America, and other nations around the world. Many European countries, I'll have a list in a moment, have eradicated BLV, but vigilance is the key there in order to keep it out of the countries that have worked so hard to eradicate it. So the work I'll present today is largely done by a graduate student, Meredith Fry, who is now off doing a postdoc at the Salk Institute, by 2 wonderful undergraduate students, Ashley Greenlick and Monica DeZuba, in my laboratory, by Professor Paul Bartlett from the College of Veterinary Medicine at Michigan State, and a DVM PhD student, Vicky Ruggiero, also in the veterinary medicine section at Michigan State University. So bovine leukemia virus, as a way of introduction, is a typical delta retrovirus. It's enveloped. It has a nucleocapsid. The outside here coating is a protein known as GP51, and it is the protein that is most visible to the immune system. Just like HIV and HTLV and other enveloped retroviruses, they're fairly wimpy, so they can be controlled or killed by most aseptic techniques. There's a nucleocapsid, and inside the nucleocapsid is a ribonucleoprotein complex. I'll talk more about that later, but the genome consists largely of RNA. Bovine leukemia virus infects primarily B cells, and once inside the B cell, it will actually integrate into the host chromosome and become part of the genetic complement of that particular B cell. There are 2 states that we usually recognize in BLV infection. One is known as aleukemic, and the other as persistent lymphocytotic. The difference is that in an aleukemic animal, it shows BLV positive by serology or even by PCR of B cells, but has a normal lymphocyte count. In persistent lymphocytotic cows, we see an expansion of the B cell component, and I have some dramatic examples of that coming up in the talk. So AL and PL would be the 2 most common disease states in BLV infection. There are times when we see no significant difference in effect between these 2 disease states, and so we combine them to just BLV plus and BLV minus, but AL and PL can be distinguished in other ways, and I'll try to bring that out as I talk. Occasionally, I should say that about 30% of the cows that are infected with BLV will develop this persistent lymphocytotic state. So it's not an insignificant number of animals. In rare cases, usually 5% or less, some people use the numbers 1 to 10% of cases, the B cells that are infected with BLV will click, and we don't understand the exact mechanisms, but they will become lymphoma cells and produce a lymphosarcoma. And it actually turns out that lymphosarcoma is now one of the major reasons for the condemnation of dairy cattle at slaughter. I think it's about 28% of condemned cattle are due to lymphosarcomas. BLV has a typical retroviral life cycle, and I'm just going to go through this in a really cursory fashion here. It fuses with a cell surface membrane. By the way, this is for HIV, which is a close cousin of BLV, and HTLV1. It binds to a cell surface receptor, it internalizes, releases the nucleocapsid inside the cell. The nucleocapsid is uncoated, and then the viral ribonucleoprotein complex uses its own reverse transcriptase to make a DNA copy of the RNA genome of the virus. That DNA copy is then integrated into the host genome, as I mentioned previously, and that's a stable integration. So the integration of BLV and other retroviruses is thought to be a semi-random event. That is, it doesn't really necessarily have particular sites in the genome where it targets, although some would say that it targets more open or active transcriptional units. We're not going to talk about this today, but retroviral integration can have serious consequences for gene expression, as you might imagine, for the host cell. Once inside the nucleus, the virus will use cellular machinery to do transcription and translation to make virus protein. The virus proteins, GP51, nucleocapsid, et cetera, all assemble on the cell membrane, and then the nucleocapsid or ribonucleoprotein complex comes together with that, and it buds the virus from the cell. So when you look at these on electron micrographs, you'll see viruses budding from the cell surface, not necessarily lysing the cell as with some other viruses. Eventually, sometimes these cells will get so full of viruses they will lyse, but generally with retroviruses, budding is the way that they produce new viruses. One of the key facets of retroviral life cycles, such as HIV, HTLV1, and BLV, is this idea of latency. That is, that this provirus goes very quiet and just sits inside this infected B cell, and it produces maybe some transcription, some transcripts and some proteins. We'll talk about that in a little bit. But it's very quiet, which makes this cell, as an infected cell, almost invisible to the immune system. So these can persist for a long, long time in the body, not being attacked by the immune system during this latent period. So here's a pretty dramatic example of what happens in a persistent lymphocytotic animal. On the y-axis here, I have percent surface IgM stained cells. That is mostly B cells. Surface IgM is expressed on most B cells, and so that's what we use as a marker for B cell characteristic in PBMCs, or peripheral blood mononuclear cells. On the x-axis, I have BLV status, and so this is BLV negative, BLV aleukemic, and persistent lymphocytotic. And what you notice here immediately is the large number of peripheral blood mononuclear cells that are staining positive as B cells. And I'm not going to talk much today about T cells, but you'll see a concomitant reduction in the number of normal T cells in the peripheral blood of these animals. So a reduced CD4, CD8 component, sometimes a reduced gamma delta T cell component as well. So these are relatively easy to detect. We're immunologists, so we use a flow cytometer and staining. You can do it by total lymphocyte counts on a CBC panel. You can use a Q-Scout machine and an IDEX machine to look at total lymphocyte counts and get the same result. That is, that the animals have an elevated lymphocyte count. So the genome of BLV is typical for a delta retrovirus. Looks almost identical to the genome for HIV and for HTLV1. And I should just, as an aside, mention that BLV serves as a really nice model for HTLV1 infection in humans. The main difference being that HTLV1 infects T cells and BLV infects B cells. So there's various structural proteins here. The LTRs, excuse me, are the major genetic factors controlling transcription of the viral genes. The GAG, POL, and ENV are structural genes and also enzymatic genes that have to do with virus assembly, virus replication, et cetera. We have the TAX, REX, R3G4 region, which is more having to do with host RNA shutoff, viral RNA transport, that sort of thing, the assembly of the ribonucleoprotein complex. And interestingly for BLV here, it was recently discovered that there are 10 BLV microRNAs that are encoded in a region here between N and R3. And so just such a small virus, about 8,000 or so nucleotides long, and it still encodes these microRNAs. I may have a chance to mention those as we go on with the talk. Now, during the latent infection that I told you about a few minutes ago, generally you would see only these microRNAs and sometimes TACs being expressed. And those are internal proteins. They're not visible to the immune system on the surface of the cell generally. So they would be still blind to the immune system. But they could have a dramatic effect on the B cells, any infected B cell, because of their propensity to regulate gene expression. So BLV is a worldwide distribution. On the top here are the countries that have eradicated BLV. This is largely done in the 70s and 80s, and they work hard to remain BL-free today. Obviously, with import of animals, they have a significant issue with surveillance and trying to make sure that BLV-positive animals don't enter the country. The same thing for many diseases. I was just speaking with one of the next speakers about Johne's disease in the Netherlands, trying to keep that out. So it's similar with BLV. A low prevalence in Ukraine, Italy, Portugal, Belarus, Latvia, Bulgaria, and Greece. And here's where the real problem is that Japan, the US, Canada, Mexico, I forgot down here, South America, all have very, very high concentrations of BLV infection. In the United States, we use the figure at 83% or so. I'll get to that in a moment. So BLV is fairly rampant in certain countries throughout the world. So in the US situation, this is where we are at currently. We have about 84% of our dairy herds that are infected with BLV, 16% BLV free. And even in beef herds, we have 39% that are infected. So this is a rampant infection in the US dairy and beef herds. What's really interesting, is that if you look at the control of BLV, and this is a study done by Rebecca Ladronka and Paul Bartlett in Michigan, despite this prevalence, look at the control. 87% of Michigan herds do not test for BLV. Only 13% routinely test for BLV. It's a simple serological assay, and yet it's just not used in the US generally. So we're now at this point where we have an 84% infection rate in dairy herds, and we'll focus on dairy for a moment, 16% uninfected. How do you control this infection? You can't really use the standard cull and slaughter routine at that level. So there's issues around how do we get this infection under control. And maybe a bigger question is, what's led to this point? Why are we here? Well, one thing, obviously, is this complacency. Number one, we don't test. So we need to encourage producers and veterinarians to start testing for BLV much more frequently and much more aggressively. That can be done by increasing economic incentives, showing that BLV actually has an economic effect on the dairy herd. There's a lot of other pressing problems. Johne's disease was a problem in the U.S., and TB is still a problem. Lots of other diseases. Mastitis is probably one of the largest concerns for dairy producers. We've heard a lot about that at this conference. There's a definite lack of funding and research on BLV. For many, many, many years in the United States, you could not get a USDA grant to work on BLV. It was just not seen as a problem. And that was largely because there's no clear indication that BLV had an impact on animal well-being or on production. Paul Bartlett at Michigan State has done quite a bit of research on this issue, and he has shown pretty conclusively that in fact in BLV-infected herds, both in Michigan and nationally, there is definitely a lack of milk or a reduction in milk production associated with BLV infection, and maybe more importantly, for my talk today, a reduction in survival of cows that are infected with BLV. So the longevity of cows is dramatically reduced in BLV-infected cows. So it is now estimated that U.S. producers absorb about $526 million a year in costs due to BLV infections. So there's a good economic incentive to start thinking about this disease in North America. However, if, like other delta retroviruses, BLV causes a relatively dramatic immune suppression, the actual cost of BLV in terms of the way the animals respond to vaccines, the way they respond to other infections within the herd, could be significantly higher. And that's kind of where my laboratory's thinking has started, was that can we show that BLV causes defects in the way the animals might respond to common vaccinations that are used in the dairy industry, and also possibly to novel antigens or new pathogens. So we know from the literature, I don't expect you to read this, I'll give you a brushstroke view of it in a moment. We know from the literature that BLV definitely has a relatively dramatic effect on the immune system in infected cows. That was clear. So you'll notice most of these papers, if you can see this at all, are from the 90s, late 80s, early to mid 90s. That's when about the funding for BLV research dried up and has just recently come back. But if you just look at the brushstroke or helicopter view of this, we have dramatic effects of BLV infection on T cells and on B cells and even on monocytes in infected cows. So we know from the literature that BLV is having impacts on these different immune cells. So what we, and if you think about this dramatic effect on the immune system in the background of the numbers of diseases that dairy cattle or beef cattle face and that we vaccinate for routinely, there's a whole list of them. And for example, I was shocked when I looked initially at the schedule of vaccines at the Michigan State University dairy, how many vaccines the average dairy cow receives by the time it actually reaches production age. So it's quite tremendous and quite expensive. So the cost in an immune suppressed animal could be quite significant. So this is probably repetitive for a lot of people in the audience, but maybe not for all. This is just a look at why do we vaccinate? What happens when we vaccinate animals? Well, what we're looking for is we're looking for these antigen-presenting cells, macrophages, neutrophils, monocytes, and B cells, to present antigen to T cells or B cells and activate them. So B cells or T cells that recognize antigens from the pathogen or from the vaccine will activate and they will proliferate. And they will begin to produce an immune response, different obviously for T cells or B cells, but they will activate, they'll grow, proliferate, and start to generate an acquired immune response. They develop during this period surface markers that we as immunologists can tag them with. And so we can kind of watch this process happen. And in this case, I use CD25 as an activation marker, that little red triangle that I've shown on these cells in my crude cartoon. They further will develop into memory cells, memory immune cells. And that's really what we want with vaccination, right? We want long-term memory cells. They're going to hang around for 3 months, 6 months, a year. In the case of some human diseases, years and years, and protect that animal or that person from that particular pathogen, whether it might be influenza, respiratory syncytial virus, etc., hepatitis B, etc. So that's kind of where we want to get to with the vaccine. Now, when these cells are activated, when you see the pathogen again, maybe for real this time, or you get a booster shot, what happens is the memory response kicks in, and this is much faster, much quicker. You have more cells responding to that particular pathogen antigen, and you get a nice bloom, and it happens much faster than this initial thing. And this data here, which was generated by Meredith Fry in my laboratory, is a beautiful example of what happens in a cow when it sees a novel antigen for the first time, right here, then develops this primary immune response I've shown here. That levels off for a while, and out at 56 days now, she comes back in, gives that animal another shot of the same antigen, and you see this really nice dramatic increase in the immune response, and this is measured by antibody production or antibody titer against that particular antigen. So a really beautiful example. You're going to see this again in a few moments, but it's a very nice example, classic example of a primary and a memory immune response. That's why we use vaccines. That's what we want. So can BLV perhaps interfere with this process? And that's one of the questions we wanted to get at. So let's focus for a minute on antibody production. So B cells are the primary cells which will lead to or antibody production. And there are several types in cows that we generally think about. We have IgM, IgG1, IgG2, and for mucosal surfaces, IgA. They all have different functions and they all have importance for different things. I've tried to highlight that here. For example, for neutralization of viruses, that's an important thing. And we do virus neutralization tests. People run these all the time for bovine viral diarrhea virus or bovine herpes virus. That's to see how good the sera from a cow might kill that particular virus. So IgG1 and IgG2 tend to predominate and be the major antibodies responsible for neutralization. Opsonization, big word, just means it coats the bacteria or the virus with antibody by binding to it. It makes it much more visible to macrophages, neutrophils, other phagocytic cells, so they can take that bacteria or virus up much easier. They have receptors on their surface for the antibodies, and so it makes it much easier for them to gobble those up. Again, IgG1, IgG2 are really important for that. Antibody-dependent cytotoxicity, dragging in a cytotoxic T cell so it can kill the infected cell. So you can't get to the pathogen, so let's kill the factory, and that would be the infected cell. It might be a macrophage, might be a T cell or a B cell. The other thing that these will do is they'll fix complement, and the complement system is one of those things that works better than my pointer right now. But complement system, can you see my pointer here? Maybe. Yes, okay. So the complement activation is a way of killing pathogens without using other immune cells, etc. Uses the complement system. Complicated system in the body. It's one of those areas of immunology that everybody wants to avoid studying. But complement fixation is largely done with IgM and IgG1. And these can also lead to opsonization, inflammation, and microbial lysis. So, what does BLV target? BLV targets the B cell. So this is the target that BLV goes after, is this B cell. And I don't know if you have a replacement pointer. So this B cell is the target for BLV. And so you can see that if BLV has an effect or a dramatic effect on this B cell and the transcription in this B cell, it can affect a wide range of immune responses. And that's kind of where we started our studies. So what we wanted to know was, does BLV infection have an effect on the response of cows to a common booster vaccine? And for this study, we used the Zoetis Bovine Gold vaccine, and given during a normal booster time. The cows that we selected, I'll talk about them later, had all seen this vaccine before when they were younger. Thank you. So we started with this, and particularly we're concerned in this study about some bacterial antigens, Leptospira, and a viral antigen, and that was the BHV1. The second question we wanted to ask is, what about a novel vaccine? What about a novel antigen? Like, let's say we couldn't go out and give cows foot and mouth disease, so let's use a novel antigen that won't affect milk production, won't affect the milk usage or the meat usage, but we can do that primary and secondary challenge on, and so we chose KLH for that. The only data we had to go on was actually from Ron Erskine and Lorraine Cerdello at Michigan State University, showing that during response to the J5 E. coli vaccine for mastitis, we could get a slightly lower response in the IgG2 production in BLV-infected cows. And that's significant because, back a couple slides ago, IgG2 is really important for opsonization and other functions that antibodies are responsible for. So the design of the Bovine booster study is shown here. The animals were bled 7 days prior to the study, and then given the Bovine booster injection. This is day 0, and then the immune cells were analyzed at that point. We also drew blood at day 7, 14, 18, and 28. So we were looking at just simply a booster response to the vaccine. We collected plasma, fresh peripheral blood mononuclear cells. We also cultured peripheral blood mononuclear cells. These were largely to look at T-cell responses and in some ways B-cell responses, usually using flow cytometry. I'm not going to have time to talk about those today. I'm going to focus here on the antibody production only, IgM, IgG1, and IgG2. So first of all, IgM. You can see pretty clearly from these graphs, and this is Meredith Fry's work, the blue is bovine leukemia virus positive cells, the green is bovine leukemia virus negative serum, sorry, not cells. The optical density here from an ELISA test is on the y-axis, and the days post-vaccination on the x. And you can see clearly, first of all, that the animals did have a really nice response to the vaccine booster. You see a nice uptick after the booster injection of the antibody titers for IgM. With Leptospira harjo, Pomona, and BHV1, we all saw the same thing. We saw the same thing for all of those. That is, that the BLV positive cows started out with a lower IgM. IgM was lower throughout the course of the vaccine booster response, and never quite caught up to that we saw with the BLV negative cows. IgG1 was not quite the same, so there was really not a big difference in IgG1 for either Leptospira harjo, Pomona, or for BHV1. And IgG2 was a little variable, and we've seen this in other cases as well. Nothing with Leptospira, no real differences, maybe a little bit at the end that more animals might tease that out. But definitely with BHV1, we saw a reduction in the production of IgG2, just as Ron Erskine and Lorraine Sardillo had to the J5 vaccine. IgG2 was lower in the BLV infected cows. So BLV was having an effect on the production of IgM and on IgG2. So just to summarize very quickly for you, BLV positive cows had less antigen reactive IgM at day 0 than BLV negative cows. That remained throughout the course of it, and the vaccine booster response showed some specificity to antigen. So KLH, so this is our new antigen. Cows have never seen KLH. It's keyhole limpet hemocyanin. It's from an aquatic organism, so unlikely a dairy cow had seen this before. This gets interesting in a moment when I show you the data. We did both B-cell immunity, T-cell immunity. I'm just again going to focus on the B-cell immunity and the antibody productions for today's talk. Our KLH injection was with a adjuvant DDA, and on day 0 we pre-bled at day 7 to look at the overall immune response or immune status of the cows, and then throughout the course we went out to 56 days, which was in our minds, our naive immunological minds, time for the immune response, the initial immune response, to wane and be low again. We then boosted with KLH plus DDA for a second memory response, and then again bled in a sequential pattern to look at the production of different antibodies. And here's that data I showed you again, that beautiful IgG1 response that Meredith saw in this study. So it's very nice. BLV minus and BLV positive animals seem to track together. But here's IgM again. IgM started out lower, and it remained lower throughout the course of the primary immune response. Once we boosted the animals for a secondary immune response, we did see them eventually catch up to the uninfected or BLV negative cows. Again, BLV positive in blue, negative in green, OD on the y-axis, and days post-injection on the x-axis. IgG2 was largely the same. It also was a beautiful immune response curve, just like IgG1, so no difference in IgG2 or IgG1 levels. I hope to come back at the end of the talk, if I don't run out of time, and talk about why there may still be a significant effect of BLV on IgG1 and IgG2, even though these graphs look very similar between the negative and positive cows. I'm going to skip that a little bit. So there was no significant difference in our IgM between the Bovishield and the KLH studies. So when you combine those and you look at day 0 levels, this is before the animals saw Bovishield boost or KLH, just animals in the herd. We bleed them, we collect plasma, we look at IgM. The overall IgM levels were lower. This is total IgM, just sera, not looking at antigen specific at all. Total IgM levels were lower in BLV positive cows than in BLV negative cows. So this caught Meredith's attention and mine. And she wanted to pursue why might that IgM level be lower. Well, one of the things that IgM, just a little more immunology here, IgM is unique in that it is actually a pentameric structure of antibody molecules, and they're all stuck together by this little chain down here called the J chain, IgJ. Interestingly enough, for those that might be interested in milk or mucosal immunity, IgA, which is more important for mucosal immunity and in milk, is also joined by the same molecule, IgJ, and it's a dimeric structure. That'll become important in a second. And so we ran another study just to look here, and in fact, we saw with a new group of cows that have never seen the Bovishield vaccine or the KLH, that IgM levels, total IgM levels, were lower. We looked at IgJ expression, and without going into a lot of details, what we did was we purified B cells from BLV positive cows and BLV negative cows using high throughput flow cytometry and cell sorting. And we looked at the gene expression for IgJ in BLV negative and BLV positive cows, and there was, in fact, a very significant reduction in the expression of the IgJ molecule in B cells from BLV positive cows, suggesting that a lack of IgJ might be one reason for the lack of IgM or the reduction in IgM expression. Data collected recently by Monica DeZuba and Ashley Greenlick in milk shows that if we look at IgA levels, the dimeric structure that also depends on IgJ in milk, and we look at it in BLV positive cows, we see a significantly lower level of IgA production in the milk from BLV positive versus BLV negative cows. So this, we've also seen this with IgM. So IgM and IgA in the milk of BLV positive cows is significantly lower than the milk from BLV negative cows. So this led Meredith onto a study of B cell development. And I'm not going to go through this in any detail for you today, obviously for time reasons, and I don't want to put you to sleep early on a Saturday morning. But the key points of this slide are that once B cells become activated, shown here, they start to express certain cell surface markers. The hope is, I showed this on the earlier slide where the cells proliferated a little bit. The hope is they'll form germinal center cells. And this is where the B cells will replicate, they'll proliferate, and you'll get that nice memory response that you're looking for with vaccination, or you're hoping for when you get the flu so you don't get it again. So germinal center development is very critical for the function of B cells. Now this is also the place where B cells, once they make antibody, they're not done. When a B cell starts to make antibody and becomes part of the germinal center, it starts to undergo a process we call somatic hypermutation. As it proliferates, the antibody molecule, the genetic structure of it, changes slightly. And what it does is it starts to get a better and better, a higher affinity antibody produced. And so as over time, as that B cell proliferates, the antibody it makes binds to the pathogen stronger and stronger and stronger. So it's a very important part of our overall immunity, these germinal center B cells that will produce the strongest antibodies possible for opsonization, neutralization, all the functions that we already talked about with B cells. So it's a really critical piece. Those cells will also become plasmablast or plasma cells. These guys we want also. Think of varicella zoster when you got the chickenpox vaccine years and years and years ago, or you had the chickenpox. And now at the age of 60, I just got approved to have my shot for shingles. All these years I've been protected against varicella zoster virus because of that shot. That's long-lived immunity. And that is, the plasma cells are responsible for that. So long-lived immunity, these produce the most antibodies. They would be called ASCs, or antigen secreting cells, and they produce the most immunity. So this process has gene expression patterns, like many things in developmental biology, that control it. PAX5 is expressed throughout B cell development. Bcell6 has its highest expression at the formation of germinal center B cells. So when B cells become germinal center, they express large amounts of Bcell6. BLIMP1 has its highest expression in the formation of these plasma cells. IgJ has its highest expression, again, in germinal center and plasma cells. What does BLV do to these? Well, it's pretty clear to see from this. PAX5, it has no effect on PAX5 expression. BLIMP1, required for plasma cell generation. Look at the BLV positive B cells relative to BLV negative B cells. Again, these are purified B cells from infected and uninfected cows. Also, look at the Bcl6 required for germinal center formation, BLV positive versus BLV negative. Significant reduction in the expression of both of these genes. So in addition to the IgJ reduction, BLV infection resulted in a dramatic reduction in the level of those key genes that are involved in B cell differentiation. And that's just shown here simply in this graphic. Loss of Bcl6, loss of BLIMP1, and also reduction in IgJ. I'm also going to show you a little piece of data, again, BLV in blue, BLV negative in green. This is time and days post-vaccination. This is, I believe, the KLH experiment. Look at we use CD45R0 on the y-axis as a measure of the immunological memory B cells, the formation of memory B cells. Look at BLV positive animals relative to BLV negative animals. A dramatic reduction in the amount of B cells that are labeled with our memory marker, our CD45R0. So overall, in immune competence in BLV positive cows, we have a reduction in effectory memory B cells, lower activation in vitro. I didn't really show that data. Lower plasma cell transcripts, lower germinal center transcripts. And so our data is now suggesting that BLV does in fact have a dramatic effect on B cell maturation, which would include germinal center formation, somatic hypermutation, and generation of long-lived antigen secreting cells or plasma cells. Okay, so we know it's a big problem. Causes immune suppression, lots of things going on with B cells, and we're going to go ahead and start to look more mechanistically at this in the future. What do we do about it? How do we control this rampant infection in the United States, Argentina, Japan, etc.? What do we do at the farm level for control? Well, this has been worked on quite nicely by Paul Bartlett and Vicky Ruggiero in a study in Michigan. This is just a pilot study, but their idea was that let's identify, and you can think back to, there's a keynote lecture on bovine viral diarrhea virus. Same thing. When you look at a herd of infected cows, you have a bunch of them that are BLV positive, some are AL, some are PL. A couple of them have a lot of BLV. We call them super shedders. What if, like with BVD control, we went after just the super shedders? So this was the idea that Paul and Vicky had. Let's take these herds, we'll do ELISA screening, we'll do lymphocyte counts. I showed you that, the large elevation in B cells. proviral load, we do that by PCR to look at the actual amount of provirus per cell in an infected cow. And then we'll use that data, the proviral load and the lymphocyte count, to direct culling decisions. So we go after only the highest super shedders in the herd. We also institute good herd management practices, single sleeves, single needles, etc. All the things that you might recommend for a blood-borne pathogen like BLV. This is what happened over a series of months in these herds. The first herd here went from 65% BLV positive to 45%. Second herd, 59% to 24%. Third herd from 64 to 13. This is pretty darn convincing data that by going after those super shedder animals, you can really reduce the overall prevalence of BLV in a herd. And again, the idea here is going to be, let's get the herds down to a level where test and cull is actually possible. So using these super shedders, we can. And so the significance of this is pretty good. I don't even know what that number would translate into, but it's pretty significant. Maybe even more important, let's look in the same study at the level of new infections in the herds. Here's before we started, or they started, 19%, 16%, 10% new infections. Every herd, all three herds involved in this study dropped dramatically in the development of new infection during the study period. So removing the highest proviral load animals in a herd plus management practices dramatically reduces the number of new infections. And when you control a pathogen, that's what you want. You want to break the cycle of new infections. And so by going after only those highest proviral load animals, we can break that cycle and we can bring down the BLV incidence on the farm, bring down the rate of new infections dramatically, and start to get to a level where in the United States, Japan, Argentina, we could actually think about some type of a control mechanism. So they have a whole list. There's a BLV website at MSU, and I'm sorry I don't have the URL here, for the recommendations for management changes that could go along with such a program as looking at the super shedders. I believe they have a couple of papers coming out in the near future on this particular aspect of BLV control, and so I would encourage you to look at them. And finally, I'd like to acknowledge the people in my lab that contributed to this work and in other labs: Ashley, Monica, Meredith, Katie, my lab manager, my graduate student, Justin, who is so patient in organizing and supervising the undergraduates. The MSU BLV group, there are a lot more people involved here, but particularly Paul Bartlett, Dan Grooms, Ron Erskine, and Vicki Ruggiero. The farms that we use for our research, and of course, our funding partners in getting this research done. So with that, I'll close, and happy to take a question. I don't know that there's much time left. Thank you.